Okay, then let's start the class. We are at the last part of the lesson. So up till now what we were discussing was we were talking about transportation in case of plants, and when we talk about transportation, major discussion has been about the translocation of water through xylem, right? So we have discussed that part. Today we are just left with the last part of the lesson, Okomandiri. So what we will say is, or what we will discuss is the role of phloem in the translocation of food, right? Xylem is a pani transfer kore. These things we have already discussed, right? But we know that phloem translocates food, and there were several experiments that were conducted to prove that statement. As of now, it's very easy to say that phloem translocates food, but at the beginning, no, they had to do certain types of experiments just to prove that statement. So translocation of food rot, there were several evidences to support phloem is a bostu to transport kore. You need certain support, right? So hekini likhi lua. Let's finish the chapter first, then we'll discuss doubts. Okay? Water potential actually hoy ki free energy, right? Water potential gradient is free. Okay, what we can do is let's finish this lesson, and we'll do the revision of the entire lesson after completion. Okay? Ekhini kori lou agote. Okay. So just to prove that phloem translocates foods, various experiments has been conducted, and out of that we talk about 3 of them, and out of the 3 we are supposed to read only number 1, right? So there were certain isotopic studies of the phloem which proved that phloem or bhitorot ki thake food thake, right? There was analysis of the phloem sap. What is sap? The cell sap, no? Eta cellor junki inside content, taka ami ki boli kau cell sap. When the phloem sap was analyzed, they could see the presence of food materials, etc., etc. But we have to talk about this ringing and gargling experiment. Out of these 3, the first one gives a basic proof that phloem ki kore food translocate kore. So what is ringing and gargling experiment likhibo lagibo. So heading koriba ringing and gargling experiment. So who was given the ringing and gargling experiment? Name of the scientist important, so you need to know this. So what he has done is, so there was a ringing and gargling experiment that was given by Hartig, and what did the experiment prove? That phloem translocate food materials. What has Hartig done? He has taken a potted plant, and from the potted plant what he has done is he has removed some tissues in the form of a ring, and that is what is called as gargling. Gargling mane ki to remove certain tissues from a plant is called as gargling. When you gargle a plant, that does not mean you cut the plant into two halves. Exactly কাটি নিদিয়া ঠিক আছে, you just remove certain tissues, and if you are talking about the tissues that has been removed, সেই tissues কিটা কি আছিলে cortex, phloem and cambium. So he has taken a potted plant, he has removed certain tissues and he has left that plant for some time, and after some time what he has seen is যোনখিনি জেগাত ring to কাটিছিলে তাৰ ওপৰত they could see a swollen area, right, and they have proved that this swollen area is because of what? It's because of the food that could not be translocated towards the root, right. So leaves bilakot ki hoise food prepare hoise, we have the phloem will translocate the food to each and every part of the plant body. So obviously the food has to come to the roots also, but since in this area these tissues has been removed, and one of the major tissues that has been removed is always phloem, right. So since it has been removed, the food could not be transported towards the root, and as a result tahate ki hol accumulate hoi swollen area form koriya. Once this experiment was set up, then they could conclude that this swollen area was because of food accumulation. Why it got accumulated? Because phloem was removed, right. And next gradually what could be seen is ei bostu to bahut deri thoi diar loge loge they could see the development of adventitious root. So if you cut the plant from this part and you plant it again, then it will grow into a new plant karon kiyo karone roots already ase, right. So yete questions ki hai, the first question that is asked is, which part of the plant body dies first because of ringing and gargling experiment? Which part of the plant body dies first if you do a ringing and gargling experiment? It is always the roots that will die first, right, because yat to food goe ase no, khali yate hai aha nahi, so it's the roots that first. Second question that is asked is, what are other tissues included in the gargling process? So you have to know the tissues, cortex, phloem and cambium. Key tissues remove korisile. Advantageous roots. Food already আছে, food already থাকি পালে অলপ দেৰি পিছত মাটিও ঢুকি পাব ন, it's a very small potted plant no, আৰু gradually তাৰ পৰা roots, fruits already food আছে, leaves বিলাকে food দি আছে, তো ultimately root system ও develop কৰি যাব কেইদিনমানৰ পিছত, and since the root has not been developed from the radicle, তাক আমি কি বুলি ক'ম, advantageous roots এ ক'ম. নহয়, ইয়াৰে যদি ৰুটটো মৰি যায় তেতিয়াহ'লে প্লাণ্টডালো মৰি যাব, কিন্তু তুমি যদি ৰুটটোতো অলৰেডি মৰি গৈছে, এইখিনিত যদি কাটি পেলাই এইটো পাৰ্ট বেলেগত প্লাণ্ট কৰা তেতিয়াহ'লে এই প্লাণ্টডাল জীয়াই থাকিব, কাৰণ এডভান্টেজেছ ৰুটছ আছে, মাটিৰ ওপৰতো এবজৰ্ব কৰিবই. Another question, tissues removed when a plant is gardened. So as I said, from this experiment or from this section, only 2 questions are asked. First, who has given the ringing and gardening experiment? Second, 3 points. First, who has given? Second, which part of the plant body dies? And third is, what are the tissues that you have removed, right? From this portion itself, a question has been asked, right? If you see in the ring of bark that we were talking about, we have written the name of the tissues, but exactly ek to report the question to ahane okoman kurai dise, right? You cannot see exact the words in the options, right? So yar bhitor answer to ki hobo? It is, yes, it is phloem to epidermis. No doubt yato phloem ase, yato phloem ase. Obviously the xylem has never been removed, right? Talking about pit to remove karone no, centermost portion to remove korile gosal nathakiboi dekhun, right? So it is always phloem to epidermis. So it's not necessary exact dibo, okoman twist kori dibo pare. So this together with various experiments prove that phloem translocates food. Next part of the lesson, we have to talk about the mechanism of translocation. Phloem is nothing but a tube-like structure. If the food needs to get transferred from one place to another, keneke jai, right? We have to talk about the mechanism. So heading koriba, mechanism of food translocation. Is the question over? Hold. Again, as far as mechanism is concerned, various theories has been proposed, but what is the one theory that we need, or one hypothesis that we need to talk about is just Munn's hypothesis, right? Various scientists were of various opinions. They said, some said that it's because of the diffusion process, food transports from one place to the other. Some others were of the opinion that it's because of facilitated diffusion, that food used to go from one place to another. Electroosmotic theory talks about the role of potassium ions, right? Next is streaming of protoplasm. We know every cell has continuity and the inside portion is called as protoplasm. Eta cell pra antu cell there is a streaming movement, no? So that is the way the food gets transported. But we need to talk about only one thing, that's Munn's hypothesis. So heading ami Munn's hypothesis kori. বাকি কিটা তো আমি বুঝিয়ে পালো ডেফিনেটলি কিছুমানে কি কৈছিলে যে ডিফিউশনৰ কাৰণে from an area of high concentration to the area of low concentration. Kisumane koisile je it's because of facilitated diffusion. Facilitated diffusion kole what we understand with the help of carrier proteins. Yes. And some said because of ions. Some said because of streaming movement of protoplasm, etc., etc. But the most accepted is Munn's hypothesis. Actually teneke pura specifically koa nai, thik ase, they were just of the experiment he kori asile, pura proof hisabe proof koribo pare nai. Okay, so Munch has conducted an experiment just to demonstrate that how does food get transported in phloem, right? Actually, what he has done first is he has done a physical experiment at the beginning, and this physical experiment has been later related with biology, right? So first of the experiment to course, it was obviously on the base, I mean he used certain things like test tubes and beakers, etc. Biological factors was not included. Jetia experiment to hoi gol tar pisot he biological factor to include kori, he tried to explain that phloem translocates food, right? Okay, in this section what we are going to learn is certain words called as, I think you have already heard about it, but even then, have you heard certain words called as sink, source, phloem loading, phloem unloading? These are the four things that we need to learn from this. Right? So we have questions keneke ahe, phloem loading kot hoy, phloem unloading kot hoy, right? Next is it a ATP required process or without ATP hoy? Movements hoy, it is because of what, etc. So these are the points that we have to study, right? So what he has done is Mancha prothomete physical experiment to silo. At a point likhi diyasun. According to him, so what did Munch say? Food translocation from one place to another. Who is responsible? Turgor pressure, right? Turgor pressure or karne eta jegar pra antu jegale food to transport kore. And plus, if you are talking about turgor pressure, we have already read the concept of turgor pressure. Turgor pressure ketia hoy when the cell absorbs water, right? So whenever the water enters inside the cell, it exerts a pressure against the cell wall, and that is what is called as turgor pressure. And what he said is because of turgor pressure, food from one place it goes to the other place. Junto place of food to synthesize, he said, that is what is called as the source, and from where the food will get transported to the next source, that is called as the sink. Jot form korise tar kami source boli kom jole jabo, that is called as the sink. And that pipe, no, you have a connection, and that pipe is nothing but the phloem. So what he has done, physical experiment jehetu korisile, just roughly at a diagram akila. Oh, jetia tumar bostutu mass flow, whenever we will use the word translocation, no, translocation word to use korile, we understand that it always occurs through mass flow. It is not only one molecule that will go at one time. Bahut bostu, bahut water, bahut food ekelege jabo. So jehetu bahut ekelege jay, so that's the concept of mass flow. Eta molecule na jay. Because xylem jetia pani absorb kore, it is not only one molecule that it absorbs, no, huge bulk of water loi, no, so that is called as mass flow. Thik teneke phloem jetia food transport koribo, it's not one molecule of food that goes to one place, right? So that is again mass flow. So phloem, conduction of phloem hok ba conduction of xylem hok, duitate keneke hoy mass flow through re hoy, ar he karone ami process to ki boli koi translocation ko, right? right? So what he has done, physical experiment কৰিছিল যিহেতু he has taken the help of certain beakers, certain test tubes, etc., funnel, etc. So what he has done is he has taken 2 beakers, তাত কি থৈছিলে, দুটা chamber থৈছিলে য'ত নেকি concentrated solution and dilute solution. If concentrated solution in water কি হ'ব, endoosmosis. So endoosmosis or karne the water will enter. If the water enters, TP will develop, and if because of this TP from here the materials go to the other way. So prothomote physical korisel tar pisot ki hoise biology lokot link korise. When he linked with biology teo ki koisile roba eto first ki boli koisile source, eto ki koisile sink, right? Later source ot ki hoy Phloem loading hoy, sink ot ki hoy Phloem unloading hoy. This is physical. Biological khetrot what he said is প্ৰথমতে physical কৰিছে তাৰপিছত biology লগত link কৰিছে. What he said, the source is the leaves, and from the leaves the food goes to the different parts of the plant body. jole food to jai gote kita part ok ami ki boli kom sink boli, jor pura food to jai, that is what is called as the source, and that is the leaves, right? So from the leaves to the different parts of the plant body keneke jai, with the help of TP or because of Turger pressure gradient. What happens in the leaves? Leaves photosynthesis hoy, the food will get synthesized, so obviously the solutes will increase. If the solutes increases, osmotic pressure will increase. If osmotic pressure increases, water potential decreases, and ultimately endoosmosis will take place. Water lobo, swell up hobo, Turger pressure hobo, force to ki hobo, from the leaves it will be pushed to the different parts of the plant body. That's all. Endoosmosis TP উপর ডিপেন্ড নকৰে, TP endoosmosis উপর ডিপেন্ড কৰিব. इसमें नहीं, that is the reason it has drawbacks, and so it is called as a hypothesis. Right? They could not give a proper explanation because in general what we know, obviously in the leaves the food is prepared. From the leaves it will go to the different parts of the plant body. For example, it goes to the roots. Roots will also have its own osmotic potential. Not necessarily it will have low osmotic potential, it might have high osmotic potential also because it's continuously absorbing water and minerals also. Right? So they could not give a proper explanation for this part. The source of sink ole goise thik ase. So with this experiment what he says, according to him, sink should be an area with low osmotic pressure. Solutes komei hobo lagibo tetiahe ei process to cholibo. Kintu that does not give a proper explanation because sink already has its own solutes, especially roots. Continuous absorption hoy ase to solutes komi joar to eku reason nai, no? Right? So that is the reason why this is a hypothesis and not a theory. It could not explain completely, right? Assumption basis of koi dise bostu to.
Food synthesis মানে লিপচার এই লিপচার পর আর কোন লিপচার জানি কি মানে সব লিপচার জানা নাই মানে পার্টিকুলার লিপচার মানে
তো সব লিপচার হ'ব যেতে ক্লোরোপ্লাস্ট থাকে মেসোফিল সেলছত
ওকে যাই যে বলে মানে ফুড সাপ্লাই টু পাই
যোনবিলাক খৰি যায়
তাই মানে পকি গছতে লাগি থাকে যে কি
পকি যায় কিহৰ কাৰণে কিবা ডেফিচিয়েন্সি আছে ফাৰ্ষ্ট সেকেন্ড হ'ব পাৰে যে the food transportation has been stopped because of that abscission layer. I have said about abscission a little bit, no? Abscisic acid হেৰি কৰে. So যেতিয়া পাতটো পৰি যাব লাগে তেতিয়াহলে ফুড সাপ্লাইটো বন্ধ হ'ব লাগিব. So food supply to he phloem or sapot ki hoy কিছুমান tissues develop কৰে, and that tissues is called as the abscission layer. Once the if this is a tube, ইয়াতে যদি কিছুমান tissues form কৰে ইয়াৰ food to না যাব, no? So ultimately this part falls off.
So phloem bidirectional করতে
bidirectional leaves or পৰা তোমাৰ roots হলেও যাব, এটা leafৰ পৰা ওপৰলৈও যাব, ন stemৰ টিপলৈকেও যাব, ন branchৰ টিপলৈকেও যাব. So it's unidirectional কেনেকৈ হ'ব? It's bidirectional. It will go down towards the root, it will go up also. Xylem is unidirectional, তলৰ পৰা ওপৰলৈ যাব, right? certain points. The questions that are asked: first, phloem loading occurs at source sink, phloem unloading occurs at source sink. Second, phloem loading is an active process and a passive process, unloading passive process, active process, translocation passive process, active process. All you need to remember is, if you are talking about loading and unloading, so both ATP lagibo. If you are talking about the transportation, that ATP nalage, it is only on the basis of key Tiger pressure, and so we have written what? It's a passive process, right? 8 word, especially active ne passive, it can be compared with the loading and unloading of goods by the help of a truck also. Suppose you need to send certain things from your house to another destination, what will you do? You will call a truck, right? And to load in the truck, you need the laborers, right? Laborers, so laborers could be considered or compared to as ATP, right? Again, it goes to the destination, it has to unload the truck, you need the laborers again. So it's again ATP, but while they are on the way, the laborers does not have any work. So it's a passive process. Just remember, but that's all. That's all. So ultimately we have talked about transportation. Oh, TP to source upor depend kore.
Source jodi mane active process hoy, TP keneke mane.
Ar source to active ketia lobole he active once it has taken tapsat to kam nai. Right? So egita points important. So we have completed the lesson except some of the points. Last or ketaman points likhe. That's all. If this is over, points fully heading koriba. Itu hol niki? Okay, while we were discussing about absorption by the root hairs, I mean active absorption or passive absorption likhisilu niki? Just see the notes whether we have written. Have we written something called as osmotic active absorption and non-osmotic active absorption? We did not write. Okay. I think we have discussed these two terms. If you are talking about absorption by the root hairs, translocation, when we were talking about long distance transport, we had to talk about long distance transport under certain points. The first point was soil water relation, second is the translocation or it's the absorption by the root hair. So, under 5 headings we have discussed that topic, and so when we were discussing about the absorption of water by the root hairs, we have used certain words, that is it could be either active absorption or it could be passive absorption. And at this time we are not talking about ATP. What we were talking about was if the root really needed water or it absorbs all by itself, it is called as active. Or it absorbs water because of some external features that is occurring in the aerial part of a plant, then it's called as passive. What I am talking about is transpiration. If the root absorbs water because of transpiration, the process is called as passive. If the root absorbs water because it really needs it, against ki diffusion, osmosis, imbibition, heibila karono karone jedi hoy, he process to kami ki boli kom active. Jedi transpiration o karone pani absorb korise, eta kami ki boli kom passive, right? Imbibition or diffusion eke boli. Imbibition or diffusion eke boli kyo kobo nwara, karon if imbibition has to occur, the surface needs to be dry. Kintu diffusion hole dry hobo lagibo boli kotha nai. So, if you are talking about active process and passive process, most common key obviously the passive process, because we have seen 96% of water absorption is because of a passive process or because of transpiration, right. But on the other hand, if you are talking about active transport, active transport or logoto duta word associated, osmotic active or non-osmotic active. So, we are going to write certain words about them. If this is over, then ekini hol niki? So root absorption by absorption of water divided into two types, passive absorption and active absorption. Active absorption boli koa loge loge generally we know that it's with the use of ATP, but even if you are talking about active, there are two types of active, osmotic active or non-osmotic active. So osmotic active or non-osmotic active or at a basic difference wa. If you are talking about osmotic, tetyahole ATP nalage. If you are talking about non-osmotic, tetyahole ATP lage, right? Keteba question ahibo pore, give an example of a non-osmotic, give an example of a osmotic active absorption. We have done all those, root pressure, guttation, bleeding, eblak ami already korisu. Yeah, generally it is seen during the night time as compared to the daytime in the herbaceous plants. Right? Herbaceous plants, if you remember, we have talked about guttation also, no? Herbaceous plants bilake ki kore, pani release kore, no? Rati hole. Remember something called as a water stomata or hydrathodes, right? So hey pani bilak je release kore, pani absorb kori pelai to release koribo. So that kuntu process dekhisu ami osmotic active absorption dekha paisu. Directly, it is on the basis of that. Ekin likha hol niki? এইটো in a leaf এইটো point নে in a leaf it increases from base to tip and from midrib to lateral sides এইটো base 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 to tip and from midrib to lateral sides midrib we understand no major vein dal lateral sides mane sides of a leaf So this is all about the lesson and finally we have completed. Thank you.